This is the U. That's the U. Okay, sorry. I didn't know for sure. Yeah. I only had to go to for 2 to 4 years. Well, you see, it depends. Oh, like what percentage? Yeah. Because you could technically be take 1 week down.
Take my head down.
Okay, so there's a pharmacy 17. Okay. And then there's pharmacy 10. The pharmacist is the one who does the whole dose. Yeah. Pharmacy 10 is the one you talk to and you fill it. Yeah. And so is that I don't know, but I'm not being a pharmacy tech because look at this. How was it going?
We couldn't see him. No, there was a shark. We were like too late. It already night.
What, sorry, I was not listening.
No, I figured. That's how you do it.
Okay, but. So I'll take that. We'll see, that's why I shouldn't have waited so long. I should have waited to watch the whole video.
Well, to be fair, my son was like, "Oh, let's go to the gym," and I'm like, "Oh, I didn't." And it was like 10.
No, they were talking about it. Did anyone go to any events? Nice. That's good to have. Anyone go to Bible? Yeah, was it informative?
Yeah, it's over there.
I know,
I guess we're interesting.
I like it. That's way more lively than my 8 a.m. slash my 8 a.m. All right, so I did pass out this sheet about registering for Organismal, if that's something that you want to register for. It's basically elaborating on what I already told you on Tuesday. So if you want to get into Organismal, you will get into Organismal, right? But right now the caps are set pretty low. I think lectures at 5, labs are at 3 seats. They are going to increase as registration goes along. And if nothing, like absolutely nothing works in your schedule when you register, then you'll register for a limbo section, and I will work with you to get you in somewhere. It's not going to be your preference, but I will work with you and do my very best. Let's see, registration is what, next Wednesday, Thursday? You all, hopefully? I think it started today, but seniors. All right. Let's see. If you're interested in research, another reminder that this informative session is tonight. It is in the conference room. That is in the student center. It's kind of in that hallway that leads to the CAF. Right? There's a lot of like board rooms along the side. It's one of those rooms. If you're wondering where the heck that is. We should do it next week. All right. Are we ready to learn a little bit more new material? Yes. Yes, all right. I'm going to wait until the chatter dies down just a little bit here. I only have like 14 slides, I think, that we need to go through. It's not a lot. And then the last half of class will be exam review. All right, so Tuesday we talked about transcription. Now we're going to talk about translation. All right, so we learned how DNA becomes RNA. Now we need to learn how RNA becomes a functional product, becomes a protein. And I like translation better. I think it's fun, but I might be a little nerdy in that aspect. All right, so if we're talking about translation, right, we're talking about the process of making a functional protein, of synthesizing a polypeptide. So remember that terminology, polypeptide is the polymer name for a protein. Right? And polypeptide are made up of amino acids. Those are the monomers. Right? Now we've already made our RNA molecule, and we made that molecule so that it can leave the nucleus and enter into the rest of the cell. So this process of translation then happens in the cytosol, right, so that the fluid surrounding the organelles, at a ribosome, right, so that would be the particular organelle that processes the mRNA. And you can see in this image here we have transcription happening in the nucleus. Right, we are making mature mRNA. Now I can tell that this is a eukaryotic cell, A, because it has a nucleus, right? But B, I also see the poly A tail and the modified guanine cap. I also know that we've gone through splicing to remove those introns. So it's a mature mRNA molecule. It will leave the nucleus, end up in the cytosol, and the ribosome will be the site that's going to read the mRNA. All right, so we're still talking about reading specific nucleotides, talking about reading RNA nucleotides. Obviously, they have stars animations here. All right, so we have our mature RNA. Right? Our mature RNA contains what we call the genetic code. Right? It's a sequence of those nucleotide bases in a mature RNA molecule. Right? So if I were to look at this RNA molecule that I have here, it's just single-stranded. I could lay out all the letters. So if I read it top to bottom here, I could say G, C, U, A, C, G, G, A, G, and so on would be the genetic code. Right, now to make sense of this genetic code, I'm actually going to read it in groups of 3 that we call codons. Right, so now I'm going to break it up and take the first 3, G, C, U, that's a codon. A, C, G, that's a codon. The next 3, G, A, G, that's a codon, and so on. Right, so I've taken a string of nucleotides, I've broken it into sets of 3 that I've called codons, and now those codons are going to code for a particular amino acid. Right, so if I look at, I have 7 different codons here, right? I could potentially be coding for 7 different amino acids. Now we'll talk about why that's likely not going to be 7 when we talk about why the genetic code is very unique. Right? But a codon is going to specify what amino acid comes in. It's also going to specify where to start and where to stop when a ribosome reads the RNA molecule. All right, so we're reading in sets of 3s. Let's practice. All right, so if we have a strand, I've written out directionality because I'm always going to write out 5 to 3 when I have one singular strand. The first thing I'm going to do is break it into my codons. Right, so I'm going to separate it out by codons. So my first codon is AUG, UCU, and then UGU would be my third codon. I'm then going to use this codon chart in order to figure out what amino acid each of my codons code for. All right, now I have shown you this chart. It's kind of like in this square format. There's other charts out there that you might Google search and it could give you a circular chart. These charts come in many shapes and formats. I don't expect you to memorize this chart. If I ask you to do something similar like this on the exam, I would either give you the chart or tell you what the codons specify for. All right, so don't worry about memorizing it. But let's look at how to read it. All right, so I have my first codon, AUG. All right, AUG, right, if I look for my first letter, which is A, my second letter is U, so that's the second column. So I'm within this box here. And now I look for my third letter of G, I see it codes for this amino acid. That shorthand is MET. Right? This is met or methionine is the full name. Right? So I know that AUG codes for methionine. And you'll also notice it's highlighted red in my chart. Right? Methionine is also the start codon. Right? It tells the ribosome this is where to start. And it encodes for an amino acid, methionine. Right? So it kind of plays double duty there. All right, I would then move on. UCU, right? So I have U is my first letter, C is my second. I'm within this box, and U is my last one. I know it encodes for SER, or serine. Right? I don't expect you to know the full name if I don't give you the full name. You can use these abbreviations. Right? So I continue doing that, right, until I have translated out all of my codons. All right, so I have methionine, I have serine. I then found that I have TYR, PHE, GLY. Now if I count out the number of codons, I have 1, 2, 3, 4, 5, 6 codons. But I've only written 5 amino acids down. Right? Because if I look at my last codon there, my UAA, and I look at my chart, right, I have UAA, it encodes for me to stop. All right, so it doesn't actually have an amino acid. It just tells this process to stop. Right? Hence, I have not written an amino acid because this does not encode for an amino acid. All right. Best way to learn this is to practice. So I want you to work through an example here.
I'm gonna write it down. Yeah.
What is THR? I'd have to look it up. Yeah, I don't even know. It probably ends the INE. Okay.
Yeah.
I'd probably relate to it. Let's see.
I also have a doubt. I would say it.
Okay, it's like this one. 42, okay.
That one would be fun, as long as you remember.
Mostly did it right. I didn't know it. Did it take any points?
Oh, yeah.
Yeah, it's just like from like prophase all the way up until.
I love it when it has my name on it. Makes me feel Thanks very much. Wow, that was about 5 seconds. My kind of
Oh, yeah, I just forgot to put it.
I have the brushes. I had an uncle that went there on a cruise.
Oh, yeah.
I don't know how that happened, but
What?
we don't really like him, so, you know.
No.
So are you gonna go?
It's fine.
Oh. Yeah.
Thank you very much.
I'm okay with saying it's late.
So he's so crazy that he wants to be.
All right, hopefully you had enough time to do this. I gave you plenty of time, I think. So if we were to do this, right, work through our problem, what is the first step that you would do?
Separate.
Separate it, yeah, separate it into codons. So what would your first codon be? I like it, we're already going to translating. So CCU, right? All right. I don't know what that was, okay. I thought it was going to be a fire alarm for a second there. I'm like, oh, great. All right, CCU, all right. And my second set, AUG. Right, I'm separating it out into groups of 3. All right, so you could have gone a couple of different ways here, right? Because the next step is to take our codons and to translate them into an amino acid. Right, now I haven't taught you this, and I did this on purpose to make you kind of think when you got here. I could do CCU, right? So CCU would be proline, or my next one is AUG, which is my start codon, methionine. Right, so you might have started with proline. You might have waited until the start codon, which was methionine. Right? I waited until the start codon. Now, there's a couple of reasons why you could have done both, and both would have been correct. Right? I could say that this is the entire set of RNA. And if it's the entire set, right, I'm not missing any nucleotides, any codons, I would have to wait until I get to the AUG to start, right? Because AUG tells me to start translating. Or I could have told you that this is just the last little segment of the RNA, meaning there was more before this, right? And then I would have started at CCU with proline, right? So AUG can either indicate to start if it's the first AUG in the mRNA sequence, but if it's the second, third, fourth AUG that you see, it still encodes for amino acid, So then we would just say it's methionine, right? Because we've already started translating. Does that make sense? So the first one we encounter, we'll say it starts here. Any second, third, or fourth one is just going to simply be methionine. We're not going to like restart.
Does that mean every protein strand starts with what AUG codes for?
Not necessarily, but that's where we start every protein synthesis at. There might be a few codons beforehand. I don't know the reason why that might be. I know that is the case sometimes, though. Yeah. Any other questions? All right, we're going to move on. We're very chatty today, so I'm going to just pause a little bit. All right. The genetic code. The genetic code is really unique and it has a lot of important properties that make it very sophisticated. It works really well. Right? Firstly, because you might have noticed, it's a little redundant. Right? So if I'm looking for the codon that encodes for proline, right? There's actually 4 different codons that encode for proline. Right? So it's redundant. That means for a codon, right? Or for an amino acid, there could be more than one codon. And that's important, right? Because if a mutation were to occur, and we're going to talk about mutations after the exam, but say CCU, right? A mutation occurred, and now it's CCC, it would still encode for the same amino acid, right? So it's redundant to prevent mutations from making mistakes that have consequences in our genetic code. It's unambiguous, meaning that a single codon is never going to encode for more than one amino acid. Right? So UUU, which is PHE here, can't remember the full name off the top of my head, right? But UUU is only ever going to encode for PHE. Right? AUG is only ever going to encode for methionine. AUG is never also going to encode for another amino acid. It's also non-overlapping, meaning we read the first set of 3, and then we read the next set of 3, and then we read the set of 3 after, right? I'm actually going to draw this as an example on the board, right? Say we have A, U, G, C, 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 G, C, A, right? It's not overlapping in that I read the first 3. Really bad bracket. The first 3 here, and then I'm going to read the next 3, and then I'm going to read the next 3 after that. If it was overlapping, we would see something like you read the first 3, then you move on to the second letter, so UGC, and then it'd be GCC, and then it'd be CCC, right? It starts to overlap. We're using a nucleotide more than once in more than one codon. Right, so that's not the case, right? You don't want to complicate things, so we're not going to complicate things, right? One set of 3, nucleotide has been used once, it's not going to be used again as a part of another codon. All right. It's also conservative, right? In the fact that if you look at a particular amino acid, right? Let's look at serine here, right? So UCU and so on. Right? If you notice for serine, the first 2 letters are all UC. Right? It's conserving the first 2 letters. It's only the 4th one that changes. Or excuse me, the 3rd one that changes. Right? But even if the 3rd one changes, it still encodes for the same amino acid. So again, if a mutation were to occur, right, and UCU became UCA, it still encodes for the same amino acid. All right, that's important. If a mutation starts to occur where it changes the amino acid, then we can have consequences for how a protein is built. All right, so it's redundant, it's unambiguous, it's non-overlapping, and it's conservative. All right, all of these make our genetic code really easy to read. It makes it more resistant to mutations and mistakes, right? And it makes it so it's unambiguous, right? A codon is a certain amino acid. All right, so we know that we go from a codon to an amino acid, but how exactly does that get us a protein? Like what is this process? Right, well, we're going to talk about that process a little bit here. And I am going to very oversimplify this process just for the sake of time. But what I do want you to know is that this process uses two different RNAs. It uses our mRNA, which is our messenger RNA. That's the RNA that we created from our DNA strand in the nucleus. It's left the nucleus and it's found the ribosome in the cytosol. It contains our codons. And we know our codons, we don't have any thymine because it's an RNA molecule. We have uracil instead. But we also use this other type of RNA that we call tRNA, right? Or transfer RNA. Right? So it's another RNA strand, but this strand is very specific in its function, and that its function is to read the codon using an anticodon that's present on the tRNA and bring in the correct amino acid. All right, so if we look at our example here, right, we have our messenger RNA in green, it's this long strand here. Our tRNAs are these squares. We see that sometimes the squares are connected to an amino acid, and sometimes they are not. Okay? When they contain an amino acid, right, they're going to be used in building a protein. All right, so we have two tRNAs here already. If we look at the next three letters, I'm at an angle here, it would be UUC, right? That's the codon. UUC, if we were to look at our codon chart, would code for the amino acid PHE. All right, so we know when it codes for PHE. The tRNA molecule has an anticodon. It's complementary to this codon. So the codon is UUC, the anticodon is AAG. Right? So this anticodon can then come in, connect with the codon with a complementary pair, and bring in the correct amino acid. Right? So if this T molecule has an anticodon of AAG, it means it's only going to bring in the amino acid PHE. Right? If it has the anticodon of UUU, like this first one here, it's only going to be able to carry the amino acid LYS, or lysine, is the full name for that one. All right, so the anticodon is very specific to what amino acid it carries, and it's the anticodon, the tRNA molecule, that brings in the correct amino acid. All right, so if we look at big picture here, right, we have our DNA strand, right, that's where we started. We transcribed our DNA strand into an mRNA strand that was able to leave the nucleus. The mRNA strand is then going to use tRNA molecules to read the mRNA, right, and this is occurring at the ribosome. The ribosome is facilitating this. Right? And the tRNA molecules carry the amino acids, which when combined together in the sequence specified by the RNA, we get a polypeptide. Right? I like this visual. I think it's a very succinct visual in connecting two more or less complicated processes here. All right. Let's talk about the tRNA molecule a little bit more. All right, so the tRNA molecule they're made according to certain sections of our DNA. So certain sections of our DNA actually encode for these tRNA molecules to be made. And we're going to look at an example of a tRNA molecule, right, that I would say carries a serine, right? So I would superscript SER. So tRNA superscript SER. That is a tRNA molecule that we will look at. If you look at the structure of a tRNA molecule, we typically say it has like a clover leaf structure. It looks like a clover with 3 leaflets to it, right? Maybe you have a better analogy for that. This is the analogy most textbooks will use. It has this clover leaf structure with these 3 loops. It's going to contain that anticodon, right? And the anticodon reads the RNA and determines which amino acid to bind to tRNA. And it's also going to have an acceptor stem. So if we look at the acceptor stem, opposite the anticodon, there's this area that is not hydrogenly bound to another side of this molecule, this RNA, right? It's open, right? And that's at the 3' end. It's a single-stranded region. That is where the amino acid is going to bind. All right, so some important structures. That's a cloverleaf structure, which in reality doesn't really look like a cloverleaf, right? In reality, if we were to look at it 3-dimensionally, it's a bit more complicated in how it's folded. But if we were to lay it out and look at how all the bonds work together, it would look like this cloverleaf structure. It has an anticodon, which reads the RNA and tells which amino acid should bind to the tRNA. And that amino acid binds to the tRNA at the acceptor stem. All right, anticodons being really key here. And there's my acceptor. Like I said, I always forget when I add in my little star animations. All right, so how do we get an amino acid to bind to the acceptor stem of our tRNA? Well, this is done with the help of an enzyme, right? In fact, a class of enzymes. So they're all very similar in what they do. They bind an amino acid to a tRNA molecule, but they're slightly different depending on what amino acid they are going to catalyze this reaction. All right, so there's one for each of our 20 different amino acids. There's going to be one different type of this aminoacetyl tRNA synthetase. All right, so if we look at how this is done, this is an energetically expensive process, right? It's actually very energetically expensive. We have our enzyme, right? So that's our large orange structure on the screen. Right? This enzyme is specific to a particular amino acid, and that amino acid is actually going to bind in one of the active sites. Now, like I said, it's very energetically expensive. We need energy. We're going to use ATP, right? So ATP is also going to bind to our enzyme. Right, so we're catalyzing the reaction to get an amino acid into a tRNA molecule. So our tRNA molecule also has to bind to our enzyme. And it's this third step here, when we have a tRNA molecule and an amino acid that is being bonded together, that uses up a lot of energy. Right? And in fact, it takes away 2 phosphate groups from ATP. So now we only have AMP. Right? The big picture uses a lot of energy. So what's left is now a tRNA molecule that has an amino acid attached. We would say it's a charged tRNA. Right? So the one with an amino acid is charged. The one without an amino acid is uncharged. Right, you could also call it an aminoacetyl tRNA, aminoacetyl and charged RNA, tRNA, they're the same thing. All right, so let's pause a little bit, and then I'm going to show you a video of how this happens to just kind of put that visual in your head. But I want to have you answer these questions. I'll give you a couple minutes here.
It's a clover, but not really.
The black lines and some of the bombs.
That looks like a
Organic.
fucked up school. Yeah, we have quads for my
Oh.
They could afford a quad, but they couldn't afford a
I went to schools where when I was little, and it was crazy. It was like, oh my god, my room is so
I went to a really good elementary school for 2 years. It was considered, like, it was really good for games. It had a long windows. And quite frankly, the craziest goddamn shit happened there. Someone got shot. And died, by the way. They died. The teacher had a seizure on the playground. Wild dogs got in. Like 8 kids broke their legs. The school caught on fire. Schools canceled because wildfires were so bad. It was really fun.
But did your library have books?
No, but we got to but we got to go on multiple day field trips. So quite frankly, I think we got that. Yeah, they made us put bananas in there. And they had to paralyze me. Paralyzing your face. I'm 8. What do you think?
I actually don't even know.
My elementary school biology department had a lot of funding at the Richmond that we got to.
They made us code.
Cody.
We had a robotic class, we had a coding class, we had to do music, we had to do
They're so fancy, these schools.
not fancy enough to afford original. It was like actual classes. They were temporary buildings.
They're gonna burn
They had
out.
life-size chess pieces though. Huge chess pieces. And no one knew how to play chess. Because we were like 8. Thanks guys, we're gonna chuck these at each other.
Yeah, it looks like sorry. The board game.
It does look like sorry report.
Is it Farkle? The one with the bubble in the middle, and you like smack it.
I don't fucking know what you're talking
Yeah.
About
Oh, yeah.
Castles and Magic. What is the game? You've never played it? No.
It's making me think your journal is really sad.
All right, let's go through this question. So what is the general shape of a tRNA molecule when it's laid flat?
I'm looking
What does it look like? Oh, it looks like a clover leaf, yes. All right, it has loops and it has a stem. All right, what would A describe? The acceptor stem, which would eventually accept the amino acid, right? And B would be the anticodon, yes. All right, so we talked about this process of how the amino acid is going to physically attach to this tRNA molecule. All right, and that's using this enzyme, aminoacetyl tRNA synthetase. Synthetase, yes. It always feels weird in my mouth. All right. What would happen if this enzyme became defective, no longer work? What would be the consequence for a cell? Yeah. Yeah, they won't be able to make protein. Right? So if this enzyme is no longer working, it means we can't put amino acids on our tRNA molecules. So we just have empty tRNA molecules once they're used, which means we can't physically build a strand of amino acids, which means we can't make a protein. All right, any questions? All right, I am going to now switch to a video because that's the last of my slides, but I want to show you a visual of how this is working.